Dear students, today we are going to talk with you about the organs of hemopoiesis and the immune system. That will be our next lecture. It is the fourth lecture, and the topic of the lecture is first, that is embryonic and post-embryonic hematopoiesis, the characteristics, organs of hematopoiesis, immunity, immune defense reactions, immunocytes, immunoglobulins, functional relations between hematopoietic and immune organs, age change, conception about the thymico-lymphatic status. So let's start. First of all, the hematopoiesis. The hematopoiesis is a prenatal and postnatal production of the blood cells. Hem, it means that blood poiesis, it means the development of the blood cells. And we can here distinguish the prenatal and postnatal hematopoiesis. The prenatal hematopoiesis, it begins in the wall of the yolk sac, which is extraembryonic organ, and the mesenchyme that covers it, it condenses, and here it forms the blood cells. But this type of blood formation is called mesoblastic phase, and it is initiated when the blood islands are, pay attention, so from the mesenchyme that covers the yolk sac, there is formed the mesenchyme cells have processes, they condense and they form blood islands here, and the cells in the blood islands will converted into the hemangioblasts under the influence of inductive influence of vascular endothelial growth factor, and then the cells which remain inside, they will become the blood cells, and the cells around the island, they will be converted into the endothelial cells forming blood vessels. These cells, the first blood cells which arise here, these are erythroblasts, and they will form nucleated red blood cells. So initial red blood cells, they will have nuclei. Then will come here, you are looking at the months of gestation and the stages of the prenatal hematopoiesis. So first stage, it is mesoblastic. Then comes hepatic stage phase that replaces the mesoblastic phase at the end of the 5th week. And after the fertilization, red blood cells here are still nucleated, and leukocytes begin to be formed at the 8th week after the fertilization. Then here we can distinguish the splenic phase that begins during the 4th month of development, and the spleen and liver continue the hematopoietic function until the parturition. Then finally comes the myeloid phase or bone marrow phase. It starts around the 6 months of development and increases in importance as the fetus reaches the parturition time. And at that time and after birth, and all of the hematopoiesis occurs in the bone marrow, and also the liver and spleen can also resume hematopoiesis if it is necessary. Then we are looking at the postnatal. Postnatal hematopoiesis begins at birth and continues throughout the individual's life and produces large amount of the blood cells each day. So each day in the body of the adult human, there is produced 10 up to 11 blood cells, and it's called physiological regeneration of the blood. And as individuals, bone marrow manufactures and replaces 1 billion blood cells every day through the process of hematopoiesis. Here we have stem cells, pluripotential hematopoietic stem cells, and they this least differentiated of the hematopoietic cells, they undergo cell division to form more differentiated cells. They will form progenitor cells, which also proliferate to form precursor cells. Stem cells and progenitor cells do not have histological characteristics that differentiate them from one another, and morphologically they are the same. precursor cells can be identified as belonging to a specific cell line. Each cell will be identified by a specific name. Some precursor cells are able to proliferate, whereas others are post-mitotic cells, even though they continue to mature to become a circulating blood cell. The process of hematopoiesis is closely controlled by the cytokines and the growth factors. Here we are looking how the blood cells are developing, and here we have to mention especially hematopoietic stem cell that will form all of the blood cells, that is pluripotential stem cell. These cells reside in the bone marrow, and they form 0.1% of the entire nucleated cells of the marrow. They resemble the lymphocytes in their structure. Also, they seldom enter the cell cycle. Occasionally, they experience sudden spurts of mitotic activity, forming more pluripotential stem cells. Also, stem cells are morphologically indistinguishable. They possess differing cell membrane markers that permit them to be recognized. So, on the surface of these cells, we have some receptors via which we can distinguish them. And they possess different cell membrane markers, and they differentiate into two categories of stem cells known as multipotential hematopoietic stem cells. They will be myeloid stem cells and lymphoid stem cells. Here we have to mention about the colony forming unit that is a granulocyte. It is a granulocyte, erythrocyte, monocyte, and megakaryocyte cells. This is colony forming unit for these those cells we can see here. That is colony forming unit for the myeloid cell lineage. All of these cells, blood cells, and also some of the connective tissue cells, they develop from these myeloid stem cell, that is multipotential stem cell. And the lymphocyte cells of the blood will have their own progenitor, that is lymphoid stem cell. It will be colony forming unit for the lymphocytes. So this is the GEMM for the megakaryocyte, erythrocyte, monocyte, granulocyte. They are responsible for the formation of progenitor cells that give rise to the myeloid cell lines. Here will be a burst forming unit, erythrocytes, that give rise to the, here we'll have colony forming unit for the erythrocyte, then colony forming unit for megakaryocytes that form platelets, then colony forming units for eosinophils that give rise to eosinophils, then for the basophils, they develop into the basophils, then for the mast cells, colony forming units for the mast cells will develop into the mast cells of the connective tissue. And here we will have colony forming unit for granulocyte and the monocytes that will give rise to neutrophils of the blood and to the monocytes. Also, some of them will form the dendritic cells. Colony forming unit lymphocyte cells, they are responsible for the formation of progenitor cells that give rise to the lymphoid cell lines. Colony forming unit lymphocyte for T lymphocytes, for the B lymphocytes, and for the natural killer cells. In contrast to stem cells and progenitor cells, precursor cells, these are precursor cells here, they cannot regenerate themselves and they cannot produce more precursor cells, but they do possess definite histological features permitting their identification as the predecessors of specific circulating blood cells. The first precursor cells of each cell lineage is, first of all, here we have to mention the proerythroblast, and here we can see proerythroblast, which give rise to erythrocytes. then megakaryoplasts, which will give rise to platelets, then myeloblasts, which will form, it is the exception to the rule because they are recognizable only as the precursors of neutrophils, eosinophils, and the basophils. So from the myeloblasts, these myeloblasts, they are recognizable only as the precursors of these neutrophils, eosinophils, and the basophils. Then we have the promonocytes. Promonocytes will develop from the monoblasts, and they will form the monocytes. Finally, they will be converted into macrophages in the tissues. And then, so we have to mention the developing cells, which will continue their functioning. But here we also have to mention about the role of the hematopoietic growth factors, that these certain cells of the body produce glycoproteins that stimulate hematopoiesis. These hematopoietic growth factors reach their target cells as endocrine hormones, and they will stimulate a specific stem cell, progenitor cell or precursor cell, to proliferate and differentiate and give rise to a certain cell type. First of all, we have to mention about the steal factor. Steal factor here will steal factor, it's very important stem cell factor. It's granulocyte, monocyte colony forming stimulating factors. Then interleukin 3, 7, they will cause the cells to enter the cell cycle and give rise to new cells here. Then we have to mention about the each of these cells will have different steps in their formation. For example, erythropoiesis, the formation of erythrocytes, requires two forms of progenitor cells to produce 2.5 upper 10 upper multiplied 10 upper 11 red blood cells on daily basis. If the number of red blood cells in blood is less than the normal amount, endothelial cells of the kidneys, peritubular capillary network, and hepatocytes of the liver release erythropoietin, and this factor will activate formation of the erythrocytes from the pro-erythroblasts. And the pro-erythroblasts then will accumulate high amount of rough endoplasmic reticulum, and it produces hemoglobin, globin moiety of hemoglobin, and the amount of hemoglobin will gradually increase in the cytoplasm of developing cells that will form basophilic erythroblasts. Then we will see polychromatophilic erythroblasts, then will be converted into the orthochromatophilic because amount of hemoglobin increases in cytoplasm. Then it leads to oxyphilic staining features of the cytoplasm that were before stained basophilically. Now we will have staining with oxyphilic and basic stains. Then the cell will extrude its nucleus, and inside of the cell will remain remnants of the organelles, and this cell will called reticulocyte. Then this network will also extruded from the cell, and it will become mature erythrocyte. Here we have to know that developing erythrocytes in the bone marrow, they form some islands, and in the center of that island we have macrophage. So around the macrophage, these are developing all of these cells. Why we need a macrophage? We know that macrophages have different functions, and one of their functions it is the phagocytosis, and also in the bone marrow they will supply iron to the developing erythrocytes. And it's also an important factor that macrophages of the bone marrow, they take up iron from the blood circulation and supply them to the erythrocyte, developing erythrocytes. Then something about the granulocytopoiesis. In the granular cytopoiesis, developing granulocytes, these are neutrophils, eosinophils, and basophils of the blood. These are granulocyte leukocytes, and they have general features of the development. Here we can see the intermediate stage via which they reach their fully mature state. The first morphologically recognizable cell of the granulocyte precursors, this is a myeloblast. These are myeloblasts here. And the second is the promyelocyte. This is promyelocytes. And neither myeloblasts nor promyelocytes possess specific granules. However, all of three granulocytes share these precursors. The next cell in the lineage has specific granules. Here we are looking that in this stage, the cells will accumulate some specific granules. And these are neutrophilic, eosinophilic, and basophilic myelocyte. So the next stage here, this is the myelocyte. And after will formed metamyelocytes, and in this stage we can recognize that this is neutrophilic metamyelocyte, eosinophilic, or the basophilic myelocyte. So here we have to remember that with differentiation the size of the cells decrease, the nucleus becomes more densely stained, and they undergo segmentation. First of all, they become like narrow rod, and then they undergo segmentation. So finally we will have segmented nucleated cells, polymorphonucleated cells. And also we have to remember that first of all in the cell appear azurophilic granules, these are lysosomes. Then in the cytoplasm will appear specific granules. And other organelles with each differentiation, these decrease in the cytoplasm. Finally, we will have mature these neutrophils, basophils, and eosinophils. The platelets are derived from the colony-forming unit for the megakaryocyte, and they enlarge by undergoing endomitosis, where cell undergoes mitosis without cytokinesis, and gives rise to very large cells known as megakaryocytes. These large cells lie next to sinusoids and extend their cytoplasm into the sinusoidal lumen. The cytoplasmic projections undergo fragmentation along demarcation channels. If we will look here, that is megakaryocyte, it's these fragments, and there are demarcation channels inside of the cytoplasm, and they release proplatelets into the sinusoids. The proplatelets disperse into individual platelets and enter the circulation. So that is a megakaryocyte. And lymphopoiesis, it is the development of the lymphocytes. They begin with a stem cell colony forming for the lymphocytes, which give rise to the progenitor cells, colony forming units for the T cells, T lymphocytes, B lymphocytes, and this give rise to the naive T cells and naive B cells. Then we are going to talk about the here we are looking at the bone marrow. We know that by the time of parturition, by the time of birth, and throughout the postnatal life, the bone marrow becomes the universal hemopoietic organ, which gives rise to all of the blood cells. Here we can see the red bone marrow in the bone. It's present in the spongy bone and at the interstices of the spongy bony substance. In the histological sections, we can see the bony trabecules here, and between them, in the interstices, there are red bone marrow. The network, the skeleton, the framework of the bone marrow is formed by the reticular tissue, reticular cells, and at the interstices between them there are located a lot of hemopoietic cells. These are developing blood cells. Also we have here sinusoidal capillaries, and in small amount the adipose cells. So the developing blood cells, that is the sinusoidal capillaries, which present here, and the reticular cells we can see here, and also the reticular fibers, macrophage cells present here, and a lot of developing blood cells, red blood cells, white blood cells. After maturation, they enter blood circulation and go to the organs. Here we can see the megakaryocyte. It's these fragments, platelets, how they enter the sinusoidal lumen. Now about the lymphoid system, about the classification of the lymphoid system. The lymphoid system protects against foreign invasions, such as micromolecules and microorganisms, and against virally altered cells. This system is composed of collections of lymphoid tissues, and there are actually we have to know that we have 3 lines of defense that protect the body. And before talking about it, we have to know the organs which protect us. These are classified as central immune organs or primary lymphoid organs. These are sites that lymphocytes generate and maturate into functional immunocompetent cells. This is the bone marrow and the thymus. The peripheral immune organs or secondary lymphoid organs, these are sites where they capture antigens, and they provide sites for the interactions of the immunocompetent cells, immune cells with the antigens. These organs are spleen, lymph nodes, mucosa-associated lymphoid tissue, and they provide differentiation of the lymphocytes. Immune system, as we know that the immune system provides the second and the third lines of defense against invading pathogens. The first line is the defense is the epithelial barriers, like skin and mucosa, which form a complete lining and covering of the body surfaces. And also normal flora, microflora, for example, that is in our skin surfaces, also in the, for example, in the organs like stomach, which have strong acid environment. All of them protect our body. This provides first line of defense. But if the physical barrier is breached by cut, tear, or abrasion, or foreign substances can penetrate through them, on that case, the second and the third lines of defense may become activated. These are the innate immune system and the adaptive immune system. The innate immune system, it is natural immune system, it is non-specific, and it is composed of the blood-borne macromolecules like complement proteins. Then it includes toll-like receptors. It includes innate immune cells like neutrophils. We have the neutrophils, then the macrophages, and also natural killer cells. These natural killer cells kill tumor cells, virally infected cells, bacteria, parasites. The adaptive immune system, it is acquired immune system. It's responsible for eliminating threats from the specific invaders. and it has ability to react against a particular component, and it improves with subsequent confrontations with it. And now let's go to the innate immune response. Here we can see the physical barriers, just I mentioned, and chemical barriers. Soluble factors, we have a lot of, they provide also innate immune response, like lysozyme, interferons, and all of these are antibacterial peptides. Here also present defensins. They synthesized and released by the complement, by the epithelial cells. They defend the body against gram-negative bacteria. Also, they serve as chemoattractants. These will serve as chemoattractants for the T lymphocytes. We have also a lot of the cytokines and interleukins, like they produced from the lymphocytes. We have chemokines, which serve as chemoattractants. And then here we have in innate immune response, so we have the main components: complement proteins, macrophage neutrophils, natural killer cells, and toll-like receptors. The macrophage cells or dendritic cells, they have receptors for antibodies, Fc fragments of the antibodies, also for complement receptors. They also have receptors that recognize carbohydrates that are not usually present on the surface of vertebrate cells. Then here we have also, this is the macrophage cell, that is a bacteria which it attaches via specific ways. Then the complement proteins, they bind the surface of invading cells, and they enhance recognition by the phagocytes. So they assist in the recognition of the bacteria by the macrophages, because these chemicals will surround the bacteria and they make these bacteria more accessible and more recognizable by the macrophages, by the phagocytes. And they also provide lysis of bacterial cells, the complement proteins. And then we have to mention about the natural killer cells. Here we have natural killer cells. They are similar to cytotoxic T cells, but they do not have specific receptors for antigens. That's why natural killer cells also belong to the innate immune response, not to adaptive immune response. Actually, but they are types of lymphocytes also. They have Fc receptors. They have Fc receptors that recognize the immunoglobulins, the portions of immunoglobulin G here for the Fc fragment of the immunoglobulin G. And that acts as signal to kill the target cell. So when the immunoglobulin G will bind to the to this NK cell, and another end of immunoglobulin G binds to the virally infected or tumor cell, so on that case the natural killer cells become activated. That is antibody-dependent cellular cytotoxicity. This mechanism is called antibody-dependent cellular cytotoxicity. And when this binds, the NK cell is activated, and it produces a lot of perforins and then granzymes that will form the pores on the membrane of the target cell, and by that way NK cell kill this tumor cell, and it activates the apoptosis. Then what I wanted to mention, it is about the toll-like receptors. Toll-like receptors, they are they are integral proteins that present in the plasma membrane of the cells of the innate immune system. For example, in neutrophils, macrophages, they function when arranged in pairs. So there are two molecules, they form pairs. They may form homodimers or heterodimers. And some of these receptors are transmembrane proteins, whereas others are associated only with the cytoplasmic aspect of the cell membrane. So they may be located in the cell membrane or inside of the cell on the membrane of intracellular components, some organelles. Almost all of the toll-like receptors induce the nuclear factor kappa B. They induce production of the nuclear factor kappa B, and that is the transcription factor that initiate intracellular response sequence, culminating in the release of specific cytokines. So when the toll-like receptor is activated, it activates some second molecules, for example, MID88, that will activate nuclear factor kappa B, and it's in turn will activate some genes that will provide production of the pro-inflammatory genes. They will form pro-inflammatory cytokines, tumor necrosis factor, interleukin-1, and so on. They will be released outside of the cell, and they will cause sepsis, they will cause inflammation. So innate immune response, it is not stable, it is dynamic and in nature, and it's capable of regulating both the inflammatory and immune responses equally. Now, when we so as we see here, toll-like receptor, it recognizes some products on the surface of the bacteria. In this case, the component of the cell wall of the bacteria, it is LPS, lipopolysaccharide, that will be recognized by the toll-like receptor, and when it binds to it, it leads finally to release of the cytokines from the immunocompetent cell. It's mainly macrophage cells. And now we are coming to the adaptive immune response. Adaptive immune response exhibits 4 distinctive properties. It is specificity, diversity, memory, and self-non-self recognition. That is the ability to distinguish between structures which belong self and to non-self. Here will participate the cells which are mainly, these are T lymphocytes and the B lymphocytes. And also we have here antigen presenting cells which will present antigens to the T lymphocytes mainly. These cells communicate with members of the innate immune system as well as with each other, not only via signaling molecules, but also by the presence of markers on the cell membranes. By the markers on the membranes, these will be their receptors. And now when we talk about the immune response, we have to mention that we mentioned that we have primary organs of the immune response and we have peripheral organs. Here we are looking at the bone marrow. Here that is schematic representation of the bone marrow and the thymus. As I mentioned, these 2 organs, these are central organs, and they provide production of the immunocompetent T and B lymphocytes. They are primary lymphoid, central lymphoid organs. And here we have to know that all cells produced in the bone marrow, but for T lymphocytes it is important that they become immunocompetent. For this reason, the B lymphocytes become immunocompetent in birds. They become in the bursa fabricius. That's why they called B lymphocytes. But in human, there is not bursa fabricius. That's why B lymphocytes become immunocompetent in the bone marrow. But T lymphocytes, they leave bone marrow and they go to the thymus, where they acquire their immunocompetency. That's why these both organs belong to central organs. Afterward, these cells leave these organs, enter the general blood circulation, and go to peripheral organs of lymphoid system, where they will undergo the second level of the education, specialization, and this second step will be called antigen dependent differentiation of T and B lymphocytes. So in bone marrow and thymus taking place antigen independent, because they still did not see antigen, but in the peripheral organs takes place antigen dependent differentiation of these T and B lymphocytes. And here we have to mention that is a spleen schematic representation, and also the lymph nodes. Here each of the lymphocytes, they have their own sites in the for example, by pink color we have sites for B lymphocytes, and by blue color we have sites for the T lymphocytes. And so they will have their own sites, and after the full differentiation they enter the blood circulation and go to the tissues, and there will participate in immunological reactions. Lymphocytes, we are looking here what is the structure. All of the cytoplasm is occupied by the large nucleus. What we have to know that each of the lymphocytes in blood, they are differ from each other via their receptors. So they these T and B lymphocytes, they are need some receptors for recognition of the antigens. It is in B lymphocyte, these are surface immunoglobulins, and in case of the T lymphocyte, they have T cell receptors. Then we have to know that so each of the lymphocyte, it has its own receptor via which it can recognize specific particular antigen. And the immune system can recognize via specific this enormous number of lymphocyte clones that are formed by a rearrangement of the 400 or so genes encoding these receptors. So we have a lot of lymphocytes, each of which have their own receptors for recognition of their own antigens. Cell surface proteins that enable lymphocytes to interact with antigens, they are membrane-bound antibodies for the B lymphocytes, and T cell receptors for the T lymphocytes. And we have to know that also the molecular structures of antibodies and T cell receptors differ. They are functionally equivalent in the ability to recognize and interact with specific epitopes. In case when into the body, for example, we have a lot of these lymphocytes. If into the body have entered some particular antigen, for example, if this has this shape, on that case we have just this cell which can bind, because as we know, ligand, antigen, and the receptor have to match each other precisely. So if the body encountered some particular antigen, there is one cell, for example, that recognizes this one, and then from this one cell it will undergo proliferation, and it gives rise to a lot of cells. They will proliferate into the effector cells and memory cells. Effector cells will provide immunological response. If this is B lymphocyte, it will produce a lot of antibodies, and also there will be formed memory cells that will keep memory about that antigen. So activated cells known as effector cells, they are responsible for carrying out an immune response. And effector cells derived from B cells, they are called plasma cells. In this case, they will form plasma cells and produce antibodies. Effector cells derived from T cells, they either secrete cytokines or destroy foreign cells or altered cells. Memory cells, similar to virgin lymphocytes, they express also receptors, and they not directly involved in the immunological response during which they are generated, but these cells live for months or years and have a much greater affinity for antigens than do the virgin lymphocytes. So they will keep the memory about that antigen that have entered into the body. Formation of memory cells after the first exposure to an antigen increases the size of the original clone. So if there were just one cell, now there are a lot of cells that can recognize this particular antigen, and that's why this process is called clonal expansion. So clonal expansion we have here. And so that in adaptive immune response, the first exposure to antigen and the second exposure, they are different in the immunological response. After the first exposure, the immunological response is not so fast, it lasts some time, but during the subsequent exposure, this secondary immune response, it begins rapidly and much more intense than the primary response. And the increased potency of the secondary reaction is due to the process of immunological memory, which is inherent to the adaptive immune system. So that's why the secondary immune response in the adaptive immunity, it's more potent, more powerful, and it directed to eliminate the threat from the body. Here we can see different cytokines and their cell origin, their target cells, their function. So this due to these cytokines, our immune cells interact with each other. Some of them produce these cells, and others have receptors for these cytokines. By that way, our immune system operates. We have to know that we have to know that these cells, immune cells, they interact with each other because of the presence of specific molecules on their surface. For example, here we can see MHC1, MHC2 molecules. MHC means major histocompatibility complexes, which are present on the surface of the cells. MHC1 is present on the surface of all nucleated cells of the human body, but MHC2 present just on the surface of immune cells, antigen presenting cells, immune cells, just. And then we have to know that T cell receptor, which is present on the surface of T cells, that is necessary for binding with antigens. It is the receptor for the T lymphocyte that via which this cell binds the antigens. According to type of antigen, there is different receptors. And the receptors on the B lymphocyte that serve for recognition of antigens on the B lymphocyte, these are surface immunoglobulins, for example, this one. They are present on the surface of B cells and necessary for recognition of antigens on the B cells. So here we can see T cell receptor, MHC1, MHC2 receptors. Besides them, on the surface of immune cells, we have also CD clusters of differentiation molecules. And here we have to know the rule that CD3 molecules will interact with T cell receptors. CD4 molecules will interact with MHC class 2nd. CD8 molecules interact with MHC class 1st molecules. MHC1 molecules, they will present on their surface the endogenous proteins that are produced inside of the cell itself. And what means antigen? We have to know that antigens, these are the molecules, actually the foreign structure that can elicit an immune response in a particular host is known as the immunogen. An antigen is a molecule that can react with an antibody, irrespective of its ability to elicit an immune response. So actually, most of the antigens, they are proteins or carbohydrates. But we have to know that the region of the antigen that reacts with the antibody or T cell receptor, it is known as its epitope or antigen determinant. So not all of the antigen is accessible for our receptors of the immune cells, just there are some fragments. For endogenous proteins, these fragments include approximately 8 amino acids. They are peptides that include 8 amino acids, and they are, after breaking down inside of the proteasomes, the organelles, the proteins are produced inside of the cell. It's breaking down, and these peptides, antigen epitopes, antigen determinants or epitopes of these endogenous proteins, they will bind to MHC1 molecules and present on the surface of these cells. So all of cells may present their endogenous antigens via their MHC1 molecules. But in the case of foreign, if the cell ingested this molecule from the outside, these are exogenous antigens. They also break down in endosomal, lysosomal compartments, and their fragments will be presented on the surface of this cell via MHC2 group of MHC2 molecules. So exogenous antigens are presented via MHC2 molecules. Here we are looking how these interactions between immune cells will take place. For example, this cell is antigen presenting cell. And what does mean antigen presenting cell? I will show you in the illustration here. Pay attention to this illustration. That is the pathogen, for example. That is our view of the immunological response, that we will have some view of this on this system. Pathogen, any pathogen that encounters our body, the first line of the defense there is bleached. So the second line of defense, the cells of innate immune response, neutrophils, natural killer cells, macrophages, and dendritic cells, they encounter this antigen. Then they swallow it, break down it into small fragments, and they present them on their surface like this cell via MHC2 because it's exogenous antigen. So the dendritic cell presents it on its surface. Then this dendritic cell, after recognition of the antigen, it moves to the peripheral lymphoid organ. It may be lymph node or spleen or mucosal-associated lymphoid tissue, for example, tonsils or Peyer's patch. So there, this antigen presenting cell presents this antigen to the nearby lymphocyte T cells, B cells, and elicit an immune response. So these cells are activated and they are converted into the effector cells and the memory cells. Effector cells, in case of the T cells, will be T killer cells, and they will provide killing of the bacteria, or one cell will kill other cell, and it will provide our cellular immunity. So cellular immunity is provided by the T cells. B cells, when they activated, they become effector cells, plasma cells, and they begin to produce antibodies. And antibodies will be produced into the blood or lymph circulation or intercellular fluid. That's why this immunity will called humoral immunity. So adaptive immunity, we have of two types, cellular immunity and humoral immunity, and they will attack this pathogen. Now we are looking how the, for example, infected cell, a virally infected cell, or it may be tumor cell, and if it is virally infected cell, it produces its proteins and its antigens of these proteins. These antigen determinants will be presented via MHC1 molecule on the surface of this virally infected cell. Then we have T cell. Via its T cell receptor, it recognizes this antigen, and this cell becomes activated. And then here we are looking at how antigen presenting cell, it swallows microbe, it's maybe bacteria for example, breaks down it into the fragments. So this will be exogenous antigen, that's why it will be presented by the MHC second molecule, and the T cell will recognize its by its T cell receptor. And now we have to know that cytotoxic T cells, they carry on the surface CD8 molecules, and T helper cells carry on the surface CD4 molecules. These are clusters of differentiation molecules. And here we are going to the also we can see how entrance of one particular antigen activates that B cells, that B cell or the receptor of which match this antigen, and proliferation of this cell in peripheral organ will create more these cells which will have the same receptor that will finally kill those cells. So first exposure to antigen creates primary response, the effector cells and memory cells. And when the secondary exposure, so we have clonal expansion, and that's why the secondary response, in secondary exposure we have more cells that can react with that particular antigen. That's why it will be more potent. Now we are that is the antibody structure. That's produced by B cells, activated B cells. Each antibody molecule has Y shape. It has heavy chains. It has light chains. Here that is antigen binding regions. These are specific. It's like keyhole. So for each key there is keyhole. So as that it's a resemblance. This is an example. So each antigen will bind to its own antibody which match with its receptor. That crystallizable region, this one is Fc fragment. For Fc fragment on the surface of antigen presenting cells, macrophage, then the dendritic cells, we have receptors for Fc fragments. When this antibody binds to its to the receptor, then that cell, that macrophage is activated, and it provides the phagocytosis. We have 5 classes, main classes of immunoglobulins or antibodies: immunoglobulin M, A, E, D, G. Each of them, they have their own functions and their locations. Now, that is immunoglobulin M. It's the first antibody that is produced on the surface of the B lymphocyte, and this is the we have to know that that is the non-activated B cells will carry them on their surface. Immunoglobulin A, it's present in all fluids that produce in the body. It's tear, it's saliva, it's gastric juice, and so on. Immunoglobulins E, IgE, it's for allergic reactions, present on the it binds to the surface of mast cells, basophils. D, it's also surface immunoglobulin that present on the surface of the B cells. And G, it's vast majority, 70% of all of the serum immunoglobulins, they provided by immunoglobulin G that provide immunity against microorganisms. In humoral immune response, what we will have B cell. We also have to know that T lymphocytes and B lymphocytes, they have some differences in which T lymphocytes, the cells which require the presentation of the antigens to these cells, so that they cannot recognize they cannot recognize the antigens by their self. That's why they require presentation to them antigens. And T lymphocytes, they are subdivided into some groups. They are T lymphocytes which are T lymphocytes we have of so T cells develop in the bone marrow and but have to enter the cortex of the thymus to express the necessary molecules to become immunocompetent. And T cells possess T cell receptors that resemble antibodies in that they have also variable regions that project into the intercellular space and binds to epitopes, to antigen determinants of the antigens. And they do not recognize epitopes until the antigen presenting cells not present them. So they require that antigen presenting cells will present them antigens. They also express cluster of differentiation molecules, proteins on their cell membrane. There are 200 different CD molecules that have been identified. The T cell receptor complex consisting of T cell receptor CD3 and either CD4 or CD8 recognizes and binds to epitopes presented by antigen presenting cells. And these T cell receptors act only in the immediate vicinity, and they ignore non-protein antigens. They recognize epitopes if only they are associated with the two classes of MHC molecules, first and the second one of antigen presenting cells. These molecules are genetically determined and are unique to each individual, characterizing the cell. MHC class 1 are on the cell membranes of nucleated cells. They will recognize and T cell receptor for of the cytotoxic T cell which have CD8. It will bind to MHC1 first complex on the surface of membranes of nucleated cells, and MHC second molecule. These are present on the antigen presenting cells, and they will bind to T cell receptors with CD4 molecules, T helper cells. And we have helper T cells with CD4, cytotoxic T cells with CD8, and we have T regulatory cells which have CD4, then T cell receptor, and they have also CD25 molecules. T cells can become activated only if they recognize not only the epitope, they not only recognize the epitope, pay attention, so they will become activated if not just their T cell receptor recognize the antigen or epitope. Here we can see, and if the T cell does not recognize the MHC molecule, if this cell will not recognize the MHC molecule via its CD molecule, it cannot mount an immune response. Therefore, the T cells are said to be MHC restricted cells. So they not just enough that they will recognize via T cell receptors these epitopes, but also there is required that CD4 molecules will recognize MHC complex. That's why they called MHC restricted cells. T lymphocytes are classified into three broad categories: naive T cells, memory T cells, and the effector T cells. Naive T cells are immunologically competent and they have not yet challenged immunologically because they are not challenged because they did not contact with the antigen. But when they are challenged, they proliferate to form memory and effector cells. Memory T cells, they are they keep memory about the cells with which they are activated. After activation, T helper cells, they produce cytokines to activate other cells which will immediately kill the antigens. So T helper cells, they control, regulate all of the immunity, not just cellular, but also humoral immunity. That's why T helper cells, they subdivided into two groups: T helper 1 that activates cellular immunity, and T helper 2 that activates humoral immunity. Here we are looking the cytotoxic T cells, how they destroy infected cells by releasing enzymes that rupture their cell membranes. First of all, we are looking here that infected cell which carry antigen. This is its self-antigen that is endogenous. That's why it's presented by MHC1 molecule, and that is T cell cytotoxic, which have CD8 on its surface. Via its T cell receptor, it recognizes the antigen that is present, antigen the epitope, which is presented by MHC1, and via its CD8 it binds to MHC1 molecule. So after it become activated, and then it produces these perforins, granzymes. Perforins form perforation on the infected cell, and granzymes will enter the cell and destroy its interior content. About the B lymphocytes, they develop and become immunocompetent in the bone marrow. These cells manufacture immunoglobulins M and immunoglobulins D antibodies that insert the Fc fragment onto the on the surface. So the antigen recognition receptor on the B cell, it is immunoglobulin M and D, and so that the FAB or antigen binding fragment projects into the external milieu. The Fc portion is affixed to the cell membrane by the two transmembrane proteins. There is immunoglobulin, there is two components that bind to its surface. Activated B cells proliferate to form plasma cells and the B memory cells. Memory cells are responsible for clonal expansion, and plasma cells manufacture immunoglobulin M and then switch to a different isotype. It may be G or others. Certain polysaccharides such as peptidoglycans or bacterial membranes are thymic independent antigens because they can initiate a humoral immune response without T cell intermediaries, and that's why there is only immunoglobulin M antibodies are produced, and the memory cells are not formed. So that is the information about the lymphocytes which participate in the immunological response. Now about the humoral immune response, we have to know that that humoral immune response, it is mediated by T helper 2 cells. So these are the cells that will control the Th2 mediated response, that is humoral immune response. And we have to know here that to mount an immune response, lymphoid cells interact with one another and examine each other's surface molecules. If the molecules of the presenter cell are not recognized, the lymphocyte to which they are presented is driven to the apoptosis. If the molecules are recognized, the lymphocyte can recognize them, become activated, and that is it means that it proliferates and differentiates, on that case. And the epitope must be, first of all, for the activation, there is epitope must be recognized, and the co-stimulatory signal that is also have to be recognized here. The co-regulatory signal, it is a second signal here. So the first signal, first step, and the second step are required. So for all thymus-dependent antigens, B cells internalize and disassemble their antigen surface immunoglobulins complex, load the MHC second molecules, and place this MHC second epitope complex on its cell membrane to present it to the Th2, T helper 2 cell. So here, first step, it's T helper 2 cell recognizes the epitope with its T cell receptor. Let's pay attention here. That is T cell receptor. It recognizes the epitope that is presented to this cell via MHC second molecule on the surface of B cell, and MHC second itself is recognized by CD4 on the surface of T helper 2 cell. So that is the first step. In second step, there is T helper cell CD40 receptor have to be bound to the CD80 molecule on the surface of the B cell, and resulting in the formation, it will so it means that signal is performed and the T helper cell is activated, and then it produces cytokines, interleukin 4, 5, 6, and 10 that will activate the B cell, and the B cell is formed, undergo division, proliferation, and form B memory cells and plasma cells. The plasma cells will produce necessary antibodies that will bind antigens and provide the activation and phagocytosis. Here we also can see the same, how that is second signal, that is the first signal, TCR with processed antigen or epitope that is presented by MHC second. CD4 binds the MHC second, so that signal is performed and the B cell is activated because CD4 cell produces these cytokines, and these cytokines activate B cell that produces these, the plasma cell and the B memory cells are formed. Now cell-mediated immune response here. The ability of the cytotoxic T cells to kill a virally transformed cell depends on the two conditions. First of all, it must receive signaling molecules from an activated T helper cell. Second, it must be bound to the same antigen-presenting cell that is in the process of activating the T helper cell. So activation of T helper 1 cell occurs when the following two steps are achieved. First of all, the antigen-presenting cell via MHC. Second, it presents antigen to the T helper 1. And T helper 1 via its T cell receptor recognizes epitope. Then its CD4 molecule binds to MHC second molecule. It is the first step. Second signal, when the B7 molecule on the surface of APC cell binds to the CD28 molecule on the surface of T helper cell. So that cell is activated. T helper cell produces tumor necrosis factor, interleukin-2, gamma interferons. They activate cytotoxic T lymphocyte. At the same time, cytotoxic T lymphocyte have to itself take information from the antigen-presenting cell. In this case, the first signal, CD8 binds MHC1, T cell receptor binds epitope, and the second signal here, B7, and interaction between the B7 and CD28. So it means the activation of the cytotoxic T lymphocyte that took information from the APC cell and also from the T helper 1 cell. Only after this cell is activated, forms T killer effector cells that produce granzymes, perforins to kill virally infected cell. Macrophage activation by the T cells. Here we can see how T helper 1 cell, it assists macrophages in killing phagocytosed bacteria. Macrophages have to be activated by T helper 1 cells before they can destroy bacteria that they phagocytosed. This process requires that first the T helper 1 cell become activated. The activated T helper 1 cell then instructs the macrophage to destroy the bacteria in its phagosomes. The activation of T helper 1 cell requires two steps. In one step, T helper 1 cell CD4 molecule and T cell receptor have to recognize the epitope MHC second complex of the macrophage. And the second signal, T helper cell activates itself by expressing interleukin-2, as we see here, and it expresses interleukin-2 receptors and releasing interleukin-2, which binds to the newly expressed receptors and induces mitotic activity of the T helper 1 cell. So T helper 1 cells undergo mitosis and their amount increases. Effector cells then produce interferon gamma to activate this macrophage. Macrophage in turn produces tumor necrosis factor and auto-activates itself. And then what we will have here, that inside of this cell is produced reactive oxygen radicals, like superoxide, hydrogen peroxide, and hypochloric acid. Then hydrolytic enzymes also activated, and they will destroy bacteria. So as we see here, the activation of phagocytosis of macrophage, it also requires activation by the T helper cells. These T helper cells are controllers, regulators, stimulators of all of the immunological reactions of the human body. Here we have different CD molecules. Where are they located, their ligands, their target cells, and their functions. Now we are coming to the lymphoid organs. Lymphoid organs are of two types, as we know. About the bone marrow we told, and now we can see that thymus. Thymus has covered by capsule from which arise septa that subdivide the organ into the cortex, lobules. Each lobule has the cortex and medulla. Medulla continues to each other in all parts of the organ, but cortexes are separated from each other. In the thymus actually takes place the that is the small endodermally derived organ located in the superior mediastinum, and it has so in the thymus taking place differentiation of T lymphocytes that come from the bone marrow. So immunocompetent T cell precursors forms a bone that from the bone marrow enter the cortex of the thymus to proliferate. That is the cortex, that is the medulla, and here they proliferate and become immunocompetent T cells. And to do this, they must contact various epithelial reticular cells which present here. Actually, the epithelial reticular cells of the thymus, they are subdivided into 6 groups, first, second, third in the cortex, and 3 of them, fourth, fifth, sixth, they are located in the medulla. And when the T lymphocytes actually they leave bloodstream at the medulla from the wall of the postcapillary venules, and they migrate these precursors that come from the bone marrow. They migrate into the outer cortex, where they are known as thymocytes. Then their receptors on the thymocyte plasma lemma receive signaling molecules from the cortical epithelial reticular cells, causing them to become committed to the T cell lineage. Thymocytes begin to express some T cell markers, for example, CD2 molecules, CD7 molecules, and CD3, and not CD4 and CD8. Therefore, they are known as double negative thymocytes. Then, as the double negative thymocytes move deeper into the cortex, they express and then suppress other proteins on the surface. These double negative thymocytes then express pre-T cell receptors on the surface, pre-T cell receptors that cause the cells to proliferate. The newly formed thymocytes express CD4 and CD8 molecules and become known as double positive cells. As we see here, they have both of them. The double positive thymocytes rearrange their genes coding for the variable regions of their T cell receptors and express a low level of the CD3 TCR complex on their surface. That double positive thymocytes then that express low levels of TCR on the surface are tested by the cortical epithelial reticular cells to see if they can recognize self-MHC, self-epitope complexes. Most double positive thymocytes, about 90%, do not recognize these complexes and are driven into apoptosis and cortical macrophage phagocytose the dead cells if they do not pass this selection. This process is called positive selection. So some double positive thymocytes, 10%, recognize these complexes and allowed to mature, express higher levels of TCR and stop expressing both CD8 and CD4 molecules. When the T cells express either CD4 and CD8, they are known as single positive thymocytes, and they leave the cortex to enter the thymic medulla. Single positive thymocytes contact medullary epithelial reticular cells, as we see here, and the dendritic cells that challenge them to see if the thymocytes recognize self-epitopes that were not presented to them in the cortex. Single positive thymocytes that would mount an attack against the self are driven into the apoptosis, and they cannot pass this selection. This one is negative selection is called. and so they are driven into the apoptosis in the medulla, and the dead cells are eliminated by medullary macrophages. That is clonal deletion. Single positive thymocytes that would not initiate an immune response against the cell are allowed to leave the thymus, and they will be CD8 or CD4 molecules, and they will enter the blood circulation, and they will go to the secondary lymphoid organs as naive T cells. So we have to know that some of the 6 type of epithelial reticulocyte that is present inside of the medulla, it forms Hassal's corpuscles. These are concentrically located, and they have also calcium deposits inside, and they increase in number with increasing of age. The Hassal's corpuscles we can see here in the medulla. And we are coming to the spleen. That is the spleen structure. Spleen, actually, it is the peripheral lymphoid organ that is covered by the capsule, and it is dense irregular collagenous connective tissue capsule that is covered by the peritoneum, a simple squamous epithelium. It is the largest lymphoid organ, and it has convex area, the hilum, and the convex side. From the connective tissue capsule arise trabecules that enter the organ. Inside of the spleen, we can distinguish 2 pulps, white pulp and the red pulp. And inside of the organ we have a network of reticular tissue, the reticular cells and reticular fibers. They form physical framework of the spleen. And white pulp and the red pulp, these are main components of the spleen. The main functions of the spleen is filtering blood and destroying senescent erythrocytes, forming T and B lymphocytes and mounting immune responses, then hematopoiesis in the fetus, and if the need arises in adults. We have to know that the white pulp, it is sheet of the T lymphocytes or PALS. These are periarterial lymphatic sheets and lymphoid nodules. So they form the white pulp of the spleen. And the red pulp, it is composed of the vascular spaces or sinusoids here, surrounded by stroma of the red pulp, the splenic cords, consisting of a network of the reticular fibers that are invested by stellate reticular cells, and they prevent the collagen fibers from contacting an extravasated blood that percolates through the interstices and precipitating the coagulation cascade. The endothelial cells of the sinusoids are unusual, and so here these are the white pulp and the red pulp of the spleen. Actually, the structure of these organs you study in your practical lessons, and the lymphoid nodules we are looking here, which have germinative center, the site for proliferation of the B lymphocytes, then the mantle region, the marginal zone, then red pulp here. And talking about the spleen, we have to know that branching of the blood vessels, the blood supply of the spleen is provided by the arteries, that the splenic artery that forms several branches before it enters the substance of the spleen at its hilum. The vessels then travel via trabecula as trabecular arteries here that provide numerous smaller branches, and then they form the a central artery. They branch into central artery, which enter the white pulp, and it is surrounded by a sheet of lymphoid T lymphocyte. They form periarterial lymphatic sheets. Then around them there are nodules which form B lymphocytes, the accumulation of the B lymphocytes, and they pass, this artery passes through the center of this nodule, through the center of the periarterial lymphatic sheet, but in the nodule it may located eccentrically. Then it forms radial branches that enter the lymphoid nodules, and then they terminate at the marginal sinuses. But artery itself branches into the it enters the red pulp, and it branches into the penicillar arterioles. Penicillar arterioles continue, and they continue into the capillaries, terminal capillaries, and terminal capillaries, they are sheeted capillaries because around them there are sheets of macrophages and lymphoid tissue. Then sheeted capillaries may continue into the sinusoids immediately. It will be closed circulation, or some of them open into the interstitial tissue of the spleen. It will be open circulation. Then the blood will be collected into the sinusoids. So in the spleen we have open and we have closed circulations. Then the blood from the sinusoids go to the veins, trabecular veins, and so on out from the organ. So here, that is the we have to take in mind that in spleen the sinusoids have specialized structure here, in which the endothelial cells elongated. They have barrel shape. They are surrounded by reticular fibers. The basal lamina is incomplete, and through the wall of these sinusoids the blood cells may freely circulate inside or outside. So the spleen, it is also depot for the blood. In red pulp there is accumulation of the blood when it is needed. Also it is cemetery for the spent erythrocytes and platelets. And we have to mention also about the marginal zone, which is at the periphery. Pay attention, the lymphoid follicles and marginal zone here, that is around the lymphoid follicles between the white pulp and the red pulp. We have these marginal zones. Its region approximately 1/10 millimeter wide, and it is the interface between white and red pulp. The cells of the marginal zone are interdigitating dendritic cells, macrophages, plasma cells, T lymphocytes, B cells, and also here present marginal sinuses. And around this region, capillaries derived from the central artery enter the red pulp. For a short distance, then recur and empty into the marginal sinuses. And that is the site of migration of these cells. Red blood cell, the lymphocytes, plasma cells, they migrate through the wall of this marginal zone. They here interdigitating dendritic cells present the MHC epitope complexes to T lymphocytes. These cells recognize thymus-independent antigens to initiate an immune response. They differentiate into plasma cells, most of which migrate to the bone marrow and make antibodies. Material that is not eliminated in the marginal zone enters the sinusoids of the red pulp to be eliminated there by the macrophages. This material includes old platelets and senescent erythrocytes. Old erythrocytes are recognized because they lose sialic acid residues and have galactose moieties on their cell membranes. So that is a symmetry for the spleen symmetry for the spent erythrocytes and platelets. Then lymph nodes, these are rounded structures, convex and concave sites. They have hilum, different afferent lymphatics that carry lymph into this organ, and internally it has subcapsular sinus, then cortical medullary sinuses, then the efferent lymphatic which carry out lymph from the organ. Inside of the parenchyma, we can distinguish cortex that include lymphoid nodules, paracortex, and medullary cords. These lymphoid follicles and medullary cords, these are B zone, and paracortex, that is T zone for the T lymphocytes. In paracortex, we have high endothelial venules that provide migration of the lymphocytes to and from the blood circulation. It's also site for the differentiation of the antigen-dependent differentiation of the T and B lymphocytes. And so that is the we are talking about the peripheral lymphoid organs, and now about the mucosa-associated lymphoid tissue. We have to know that the mucosa of the respiratory, digestive, and urinary systems, they display non-encapsulated clusters of lymphoid nodules and lymphocyte infiltrations known as MALT, that is mucosa-associated lymphoid tissue. Examples are gut-associated lymphoid tissue, that is, pay attention, in the wall of the intestines we have lymphoid nodules. They form gut-associated lymphoid tissue. They present along the alimentary canal. Then we have bronchus-associated lymphoid tissue, BALT, it's in the respiratory system, and we have tonsils. All of them include these lymphoid follicles located all along the mucosa of the alimentary canal, and in the alimentary canal we have also Peyer's patches and Peyer patches that include a lot of lymphoid follicles. They also provide immunological defense, and here we can see the bacteria, how they encounter. Here we have M cells, microfold cells, that carry this information about this bacteria to the underlying macrophage, dendritic cells, and they are antigen presenting cells. Then they will present antigens to TB lymphocytes. They will migrate to the regional lymph nodes to provide immunological defense. Here we have also T regulatory cells. Plasma cells produce antibodies, immunoglobulin A, that will enter the lumen, and that is secretory immunoglobulin A that will provide defense. And the tonsils here are collections of partially encapsulated lymphoid nodules. The palatine, pharyngeal, lingual, and numerous very small tonsils we have. They are located at the entrance of the oral pharynx, protecting it from inhaled antigens. In the presence of antigenic challenge, lymphocytes become activated and proliferate, enlarging the affected tonsil. Here we have these lymphoid follicles. Each of them, they are sites for proliferation of B lymphocytes, and tonsils, they covered by, if it's palatine, it's covered by stratified squamous epithelium, and that is a connective tissue. This epithelium becomes infiltrated with lymphocytes. This infiltrated tissue is site for proliferation of the T lymphocytes. So the tonsils will also provide protection of the body from the foreign invaders. So that's all for today's lecture. See you on next lectures. Thank you for attention.